Hello students, welcome to the biology class. Today we are going to start with our very important unit, biotechnology. This unit consists of two chapters, that is biotechnology, principle and processes, and the second chapter is biotechnology and its applications. So today we are going to start with our first chapter of this unit, that is biotechnology, principles and processes. So first we will discuss what is biotechnology. As the name indicates, biotechnology that deals with microorganisms, plants, animal cells. Means it is that branch of science which deals with the living organism. It may be microorganisms, plants, animal cells, their enzymes, their strains to produce products and processes which are useful to the human beings or which are beneficial for mankind. The term biotechnology was given by the Carl Erecke in 1919. According to the European Federation of Biotechnology, that is EFB, that is our expanded form of European Federation of Biotechnology, EFB, that is biotechnology is the integration of natural science and organism, cell, part thereof, and the molecular analogues for products and the services. Means in simply we can say the biotechnology is the integration of all the natural sciences, all the living organism, and they produce the products which are useful for the mankind. So first we are going to discuss the principles of biotechnology, that while performing the experiments, while producing the various products, we have to follow some techniques. And the two main core techniques that develop the modern biotechnology. Modern biotechnology means in which we used to do some manipulations, whereas old biotechnology in which naturally we are using the living products to give the new products are there. For example, we are using the lactobacillus to form the curd. We are using the various parts of the microorganisms to form our food products like idli and something. These are known as your old biotechnologies, whereas the modern biotechnology that deals with the manipulations of the genes. So the first principle or the technique of the modern biotechnology is the genetic engineering, which is modification of chemical nature of DNA, RNA, and their introduction into another host organism to change the phenotype character of the host. Means by genetic engineering means that we are changing, we are modifying the modification of chemical nature of our nucleic acid, that is DNA and RNA, and after modification in DNA and RNA we are introducing into the another host cells to change the external appearance of the host. The second technique is sterilization method to maintain the growth and the manipulation of only the desired microbes or cell in large quantities for manufacture of biotechnological products like antibiotics, vaccine and enzymes. So the basic steps in genetic engineering. So first is the identification of desirable gene. Number two is introduction of DNA into the host to form the recombinant DNA. Maintenance of the DNA in the host, that is a sterilization method, and gene cloning, and then there is a gene transfer. So these are the various steps by which the modification of the chemical nature of DNA and RNA takes place. So in 1972, the two very famous biochemists, that is Stanley Cohen and Herbert Boyer, they constructed the first recombinant DNA. So how they did it? So the Herbert Boyer, they worked on restriction enzyme of E. coli. What are the restriction enzyme? Which cut DNA in a particular fashion and produce the sticky ends on both the strands. And these restricted ends are ligated with desired pieces of DNA. And the Stanley, they studied the plasmid DNA floating freely in the cytoplasm of bacterial cell. So this is known as the extra chromosomal DNA, circular DNA in bacterial cell or in the prokaryotic cell along with the nuclear material of bacterial cells. He developed a method of removing plasmid from the cell and reinserting into the another cell. They isolate antibiotic resistant gene from plasmid of bacteria, then linked the gene with plasmid and incorporated into the E. coli, where it could replicate using the use of new host DNA polymerase enzyme and make the multiple copies. So the steps which are being carried out for constructing the first recombinant DNA are gene encoding the antibiotic resistance in the native plasmid of Salmonella typhi, identified. What are plasmid? They are the autonomously means which can replicate by themselves circular extra chromosomal DNA. So the next is the desired DNA was cut at the specific location by the restriction enzymes, and the cut DNA was linked to the plasmid DNA and transferred into the E. coli for gene multiplication. So these are the basic steps which we have to follow. So the next is your tools of recombinant DNA technology, which we will use one by one and we will study the details of that tools. That is the restriction enzyme. So first tool we are having here the restriction enzyme. The second is a polymerase enzyme. Then we can have a ligases. So restriction enzyme, polymerase enzymes and ligases, they all comes under the heading of enzymes. So these three enzymes plays a very important role in constructing the recombinant DNA or to perform the various experiments of the biotechnology, very commonly without which the recombinant DNA, the gene transfer is not possible. That is the vectors. So what are the vectors? They are the carriers, they are the vehicles which can transfer the desirable gene into the host cell, and the last we need is the host organism or the cell, which may be bacterial cell, which may be plant, or which may be animal. So this is the introduction of your biotechnology, in which we studied about the core techniques, the definitions of biotechnology, about the old biotechnology, modern biotechnology, the principles and the steps we use, and who constructed the first recombinant DNA, that is your Cohen and the Breuer, and they have taken the Salmonella typhi as their experimental material, from which they have taken out the Salmonella typhi. So the native plasmid was of Salmonella typhium, which caused the typhoid. So this is the first bacteria which is being used. So what are the plasmid? It is the extra chromosomal circular DNA present in the prokaryotes, and the tools of the recombinant DNA technology that we will discuss one by one in our next session. Till then, thank you.